Okay, so I see we have a few people here. So welcome everybody who signed up for today's webinar. My name is Adrian Cordes. I'm an account manager in Germany, and I will host this session today, this webinar. And today with me is Arman Mehra, who's from our product marketing team. He will tell you today something about solutions for epigenetics research. Very interesting topic for today. So to guide you through, I will give a quick overview about Novogene, who we are, what we can do, very briefly. And then we can switch over to Arman, who will give you some more technical insights in epigenetics. Novogene was founded in 2011 and is growing since then as the interest in NGS solutions and sequencing solutions is increasingly being used in research and many other fields. So I know it from my own experience when I did my PhD and postdoc. Using sequencing solution is a bigger part now in research, and we are proud to offer these solutions on a globally footprint. So you can see here on this map, we have sequencing labs in the US, we have sequencing labs in Europe. I will give you some more insights in a minute. And we have sequencing labs in whole Asia. So we are proud to offer really good quality and good service all over the globe. So no matter where you are, we can offer that. To focus a little bit now on the Europe side, we have now 2 sequencing labs opened. So the first one was in Cambridge in the UK with the state-of-the-art sequencers and sequencing solutions, also offering high automation system with our Falcon system. And we are very proud that from beginning of this year, we can also offer our sequencing solution from the EU in Germany. We have a new sequencing lab in Munich. Just a quick overview of the services we can offer. Of course, we will focus today on epigenomics, but just for you to know, we have a lot more in place. So when it comes to human genome, we can offer everything what's possible, like whole genome sequencing, whole exome sequencing, for example. Of course, very popular is the transcriptomic sequencing for mRNA, of course, but there's also a lot more like non-coding RNA, single cell, etc. We can offer services and sequencing solutions for the whole microbial world, let's say, for resequencing, metagenomic sequencing, etc. Epigenomics, of course, we will talk about today. And of course, no matter which species you are talking about and doing research on, we can sequence all animals and plant genomes, of course. Just a few points I want to mention. Why you should choose Novogene. What's important to us, of course, to offer reliable results. And that's a big point, and to give you really good quality data. And this is also, we can do that due to our large capacity with automation system and high throughput sequencing. So we see barely batch effects through our automation system, so that we can offer for you. We have always the latest technology in place for Illumina, PacBio, and Oxford sequencing. I will also give you an insight on the next slide about that. One point which is also very important for many people, many researchers, and also from my own experience, is the turnaround time. So when we can deliver you the sequencing results. I worked with core units in my time in research and sometimes waited for, I don't know, month, let's say. So we know that's important, and we do everything what's possible to give you the data very fast. If you are not into bioinformatics, we have experts and very experienced people in-house who can help you with that with nearly all our services. So reach out to us if, of course, you need help with that in your project. Yeah, these are the latest sequencing platforms from Illumina, PacBio, and Nanopore. Just to mention two of them because they were really recently, or like not very recently, but quite new. From Illumina, we have the new NovaSeq X Plus in place, and we have them in many of our sequencing labs now. And also mentioning the PacBio older one, the Sickle 2, but also the new PacBio Revio. But of course, also the newest generation of nanopore sequencing. Then one point and one thing to this to give you some insights is our sales team in Europe. We know that sequencing solutions and sequencing projects are highly individual and sometimes very complicated. So what's really important for Novogene and for us to be there for you. So we established territories in Europe to be very locally at your place. So we have representatives and contact persons which are near to you and can speak your language. So we understand that there's sometimes a high support level needed, and we really want to match that. So you can reach out to us if you have any questions to design your projects to in the end give you the data you really need. And we can do it. We can visit you at your office. We can do online meetings. We can everything you are interested in. And just for you to see who's our team. So it's not an email. It's not only the name. So there's a person behind, and here you can just see the big team in Europe. So we have people in the UK, in Ireland. We have our DACH team, and we have people in Southern Europe. Our Nordics team, Eastern Europe, and Benelux. And yes, as I mentioned, we are there for you. And with that, I'm already finished with the introduction, and I will hand over to Arman.
Thank you, Adrian, for the introduction. I'll share my screen now. Can you see the full screen?
Yes.
Perfect. So we are ready to go. Good morning, good afternoon, good evening, wherever in the world you're joining from. I'm Arman, and I'm a product marketing manager with Novogene Europe. And today I'm going to talk about next generation sequencing solutions for epigenetics research. Now, this webinar will give you a quick overview. So we won't be going into many details. We have about 30 minutes. So we'll go through some of the solutions for methylation sequencing, like Bisulphite sequencing or the enzymatic metal sequencing. We'll talk a bit about ChIP-seq, RIP-seq, ATAC-seq, and also a short introduction on sequencing of non-coding RNAs. So let's begin with the introduction and to talk about what is epigenetics. So epigenetics is the study of how organisms control gene activity or regulate gene activity without changing the DNA sequence. So these epigenetic mechanisms, they allow the cells or the organisms to respond to their environment by altering their phenotype without a change in genotype. These epigenetic modifications can be inherited and can also be reversed. And as we all know, that epigenetics, it plays a key role in development, aging, health, disease, and several other biological processes. So here you see this diagram of some of the epigenetic modifications, which could be DNA modifications or chromatin modifications, RNA modifications, and so on. So I'll first start with DNA modifications and mainly focusing on DNA methylation. So DNA methylation is the process by which methyl groups or CH3 groups are added to the DNA molecule. So usually adenines and cytosines can be methylated. As far as eukaryotes are concerned, and even among eukaryotes, I would say mammals, So this methylation typically occurs in cytosine and is found in the CpG context, which is cytosine guanine dinucleotide context. In other organisms, such as plants, DNA methylation can also be observed in non-CpG contexts. And when located in a gene promoter, DNA methylation generally, it acts to repress gene transcription. Now, it's important to study DNA methylation because it helps us to address several kinds of research questions. So plant and animal researchers can use it to study development, behavior, environmental resistance, agronomic traits, and so on. While clinical researchers can study tumors, molecular markers, and pathological mechanisms. So another important point here is to talk about which sequencing platforms can be used for epigenetics research, but particularly for studying DNA methylation. So at Novogene, we offer 3 platforms: the Illumina, PacBio, and Oxford Nanopore. Here you can see the NovaSeq X Plus from Illumina. Here is the PacBio Revio, their latest platform. And from Oxford Nanopore, we have the PromethION platform. So today's webinar will focus on Illumina-based applications. However, I would like to mention that PacBio and Oxford Nanopore, they have the advantage of allowing direct detection of DNA methylation. But at least for today's webinar, I'll focus mainly on Illumina-based applications. So let us begin with methylation sequencing, starting from whole genome bisulfite sequencing. So whole genome bisulfite sequencing is a powerful technique which is used in genomics and epigenomics to study DNA methylation, and that too in a genome-wide scale. So it provides high-resolution information about the methylation status of single cytosine nucleotides. So here I'd again like to emphasize that methylation can be in adenines or cytosines, but when we are talking about bisulfite sequencing, we're talking about cytosine methylation, and that too primarily in the 5-MC context. So that's very important to keep in mind. Bisulfite sequencing is not going to detect the adenine methylation. So what happens in this technique is that the DNA sample, it's treated with bisulfite, which is a chemical reagent. And this chemical treatment, it deaminates the unmethylated cytosines to uracil, while the methylated cytosines, they remain unchanged. And then when we do the PCR amplification, the uracil changes to thymine, while the methylated cytosine remains unchanged. So we can then do the sequencing on Illumina platforms, and in the data analysis stage, we can make out which cytosines were methylated. So there are different library preparation methods for bisulfite sequencing. So there are 2 major methods. One is the traditional method, wherein the bisulfite conversion happens after library preparation. So this method suffers from the problem of broken library molecules, and it also requires higher amount of sample. So at Novogene, we use the PB80 method, which is the post-bisulfite adapter tagging method. Here, bisulfite conversion is carried out first, and then we do the library preparation. Now, the advantage of this method is that you can work with a lower amount of sample, and it's also beneficial for processing some challenging samples like FFPE DNA or circulating free DNA. The sequencing strategy that we use at Novogene is pair N150 by default, and we also run a control to to check the bisulfite conversion rate. Now let's move on to another technique, which is reduced representation bisulfite sequencing. It is called reduced representation because here we focus on a particular part of the genome, and we are focusing on the CPG rich genomic regions. And we can enrich these regions by using certain enzymes. So at Novogene, we use the MSP1 enzyme, which cuts the DNA at specific locations at the CCGG points. So the DNA is digested with this restriction enzyme, and then we do the bisulfite conversion, library preparation, and sequencing. So this technique helps us to focus on the CPG-rich regions, unlike bisulfite sequencing, where you study methylation in a genome-wide scale. But as we know, in mammals, for example, humans, most of the methylation happens in the CPG context. Hence, RRBS is quite powerful and quite useful for pilot studies or for clinical samples and so on. Now, I would also like to mention that at Novogene, we use MSP1 enzyme, which works for several species like humans, mouse, bovine samples, and so on. It's also possible to use other enzymes if you're working with other organisms. Now I'd move on to enzymatic methyl sequencing, which was introduced rather recently, only a few years ago. This technique is different from the previous two because it relies on the use of specific enzymes that can help to distinguish between methylated and unmethylated cytosine residues in the DNA sequence. Now, because this technique relies on enzymes, it is milder. It is less harsh on the DNA compared to bisulfite treatment. So the principle here is that we use two enzymatic steps. So in the first enzymatic step, we treat the DNA with an enzyme called TET2, and also an oxidation enhancer is used. So these enzymes, they oxidize the methylated cytosines, and they protect this from subsequent deamination. Now, in the second step, we treat the DNA with the APOBEC enzyme. Now, this APOBEC enzyme deaminates unmethylated cytosines to uracil, while the methylated cytosines are protected from deamination. So they stay cytosine. And then we do the PCR amplification, the uracil changes to thymine, while the methylated cytosines are protected. So this slide gives you a comparison of bisulfite sequencing and enzymatic methyl sequencing. So as you can see here, that at the end, we get the same sequence. Because in both of these techniques, the methylated cytosine is protected, the unmethylated cytosine is converted into uracil and then thymine. So the end sequence that you get is the same, which means that we can use the same bioinformatics pipelines to analyze both of these data sets. So enzymatic methyl sequencing has several advantages. Because we are using the enzymatic treatment, it minimizes DNA damage. Thus, you can produce higher quality libraries. And as I mentioned in the previous slide, it results in the same converted sequencing, converted sequence as bisulfite sequencing. So you can use the same analysis pipelines. EM-seq can also be used for diverse samples, such as FFPE, circulating free DNA, circulating tumor DNA. Compared to bisulfite, it has superior uniformity and reproducibility of GC coverage. So we see in this particular diagram that there is uniformity in the GC content, while in bisulfite sequencing, we see biases. Now, also the library, they show uniformity in the coverage of all dinucleotides. So it is a pretty powerful technique for methylation sequencing. Now let's come to the bioinformatics data analysis. So like I said, that for both enzymatic methyl sequencing and bisulfite sequencing, we can use the same analysis pipeline because they produce the same converted sequence. So the sequencing data, it passes through data quality control. Then we do something known as the Bismarck alignment, where we use a program called Bismarck to map the bisulfite treated sequencing reads to the genome of interest. And then we do the methylation calls. So we call methylated cytosines. We are basically analyzing which regions of the genome they're located in. And then if you have number of samples or conditions or groups, we can also analyze differentially methylated regions or differentially methylated positions between the different conditions or samples. And finally, we can do some functional analysis to identify pathways that might be possibly affected. So finally, to wrap up the methylation part, I would like to give a comparison of the three techniques that we've covered so far. So in terms of principle, enzymatic methyl sequencing, it uses enzymatic conversion, while bisulfite sequencing or reduced representation bisulfite sequencing, they rely on bisulfite conversion. So a chemical treatment is carried out. In terms of genome coverage, enzymatic methyl-seq and bisulfite sequencing, they give whole genome coverage, while with reduced representation bisulfite sequencing, because we are enriching for CpG-rich regions, so it gives you reduced representation. The input DNA requirements with enzymatic methyl sequencing, it's possible to go to even lower input DNA compared to bisulfite and RRBS. In terms of resolution, all of them offer single nucleotide resolution. DNA damage is minimal in enzymatic methyl sequencing, but is high when it comes to bisulfite treatment. Finally, in terms of advantages, I already highlighted the advantages for enzymatic methyl-seq. It leads to less DNA damage. It's great for precious samples, or if you have lower amount of input DNA, the libraries have less bias. As far as bisulfite sequencing is concerned, this technique is extremely popular and established and is known to be the gold standard for methylation sequencing. And RRBS, it's a targeted approach. So it's useful in some applications like some pilot studies, clinical applications, or if you just want to focus on the CpG regions. Now, finally, target organisms. So enzymatic methyl-seq primarily targets the 5MC, 5HMC. So it's great for eukaryotes, but there is also a recent publication that shows that it can also detect 4MC, which means that we can also apply it to microbes. Bisulfite sequencing, again, for eukaryotes primarily. It is possible to detect 4MC, but that requires additional conversion steps. So you may need to make tweaks to the protocol to do so. Then RRBS. So primarily, at least at Novogene, we offer it for eukaryotes. We use the MSP1 enzyme, which works for several different species. But if you are working with another animal species or another plant species, do reach out to our account manager to see and to evaluate if we can offer it for you. So now I'll move on to ChIP-seq and RIP-seq. So these two techniques help to study nucleic acid and protein complexes. And these macromolecular complexes are important, for example, for regulating gene expression. So we all know about DNA being bound to histones, forming chromatin, and that is extremely important for gene expression. So as far as ChIP-seq is concerned, the full form is chromatin immunoprecipitation. Here, we are trying to study protein-DNA interaction, primarily within the context of chromatin. What we do here is that we first perform a cross-linking steps. So the DNA is cross-linked with the protein, and the idea here is to preserve these protein-DNA interactions. Then we do the chromatin isolation. We fragment the chromatin into smaller fragments, and then we carry out immunoprecipitation. In the immunoprecipitation step, antibodies which are specific to the protein of interest, for example, it could be a histone protein or a transcription factor, they are used to selectively precipitate out these particular complexes. Then the purified complexes can be isolated, the protein can be removed, the DNA can be extracted, purified, and finally we can do the library preparation followed by sequencing. So this technique is helpful to investigate which regions of DNA bind to a specific protein. The protein could be histones. In that case, we often call it a histone chip. It could also be transcription factor. In this case, we often call it non-histone chip. Then there are also several variations of this technique. For example, we have cut and run, cut and tag. They have different experimental protocols, but due to lack of time, I cannot cover them in detail. Also, I'd really like to highlight here that for ChIP-seq experiments, it's important to have appropriate controls, for example, input controls, IgG controls, and so on. Now, let's move on to RIP-seq. So in terms of the experimental pipeline, it is similar to ChIP-seq, but here we are trying to map the sites at which proteins are bound to RNA within these RNA protein complexes. So again, we have the RNA protein complex here. We have specific antibodies to precipitate these complexes. The RNA which is outside these complexes is digested. We can finally isolate the RNA that was bound to the protein, convert it into cDNA, do the library preparation, followed by sequencing. So it is helpful to map then the protein RNA complexes or the sites on the RNA where the protein is bound. Again, here there are several variations of the technique, such as ChIP-seq, par-ChIP-seq, but again, due to paucity of time, I cannot cover them. A couple of notes here. If, for example, you're sending your samples to Novogene for sequencing, I would like to highlight that it's important to perform ribosomal RNA depletion, post-RNA extraction, and prior to library preparation if you're expecting ribosomal RNA contamination. And then again here, appropriate controls such as input control will be required. Finally, just to wrap up the RIP-seq and ChIP-seq part, RIP-seq is for protein RNA interactions, ChIP-seq is for protein DNA interactions. They all have a number of applications, and carrying out ChIP-seq or RIP-seq helps us to understand several biological processes, which includes gene regulation, checkpoint regulation, DNA repair, DNA synthesis, and so on. Now, let's come to the bioinformatics analysis for ChIP-seq and RIP-seq. So here, first, again, the sequencing data, it passes through a quality control check. So here we are trying to assess the quality of the sequencing reads, which bases have low quality scores, is there any adapter contamination, and all of this can be removed or filtered out. Then we map the sequences to the reference genome, and then we do the peak calling, which basically means to identify regions of the genome which are enriched with these aligned reads. And these regions would then indicate protein DNA or protein RNA binding sites. Then we can also do differential peak analysis and functional analysis of the associated genes. moving on to another technique, which is ATAC-seq. It stands for Assay for Transposase Accessible Chromatin Using Sequencing. So it's a powerful technique that helps us to study open chromatin, or in other words, to assess genome-wide chromatin accessibility. So this technique, in terms of principle, it uses this TN5 transposase, which preferentially adds sequencing adapters to the open chromatin. So the regions of open chromatin are more accessible because they're not tightly bound to these chromatin-associated proteins. So this enzyme can target these regions. It can add these adapters, and these inserted adapters then allow us to amplify these sequences and then carry out the sequencing on our platform. And this then provides us with a snapshot of the accessible regions in the genome. Another benefit of this technique is that it can be performed on both bulk cell populations and also on single cells. As far as Novogene is concerned, we accept pre-made libraries for ATAC-seq. I'll move on to the bioinformatics analysis for ATAC-seq. So again, we have the raw data from the sequencing platform, and this data undergoes quality check to obtain the clean data. And then these reads, they're aligned or mapped to the reference genome. We identify regions of open chromatin using peak calling tools. We can also do annotation of the peaks with genomic features. For example, is it a promoter or an enhancer? And finally, functional analysis can also be performed. So after covering all these different techniques, I'd move on to the last one, which is to sequence non-coding RNAs. Now, non-coding RNAs are also important in epigenetics. They play a key role in epigenetic control. They can regulate gene expression by modulating transcriptional processes. They have been implicated in genomic imprinting. They can guide DNA methylation machinery to specific genomic regions, thus leading to either targeted gene silencing or suppression. They can also affect chromatin structure and accessibility by recruiting these chromatin modifying complexes to specific genomic regions. So they have many diverse functions. And as expected in living organisms, we also have many diverse kinds of non-coding RNAs. So it's a big set. So if we look at the RNAs in a typical mammalian cell, most of the RNA is the ribosomal RNA, 80 to 90%, followed by transfer RNA, then messenger RNA about 3 to 7%. So these non-coding RNAs are often 1 to 2%, sometimes even lower, but they have many, many important functions, as we also discussed in the previous slide. And they can be of different types, like long non-coding RNAs, which can be greater than 200 nucleotides. We can have short non-coding RNAs, like microRNAs, small interfering RNAs, circular RNAs. And with research, we are constantly discovering new species. So in terms of sequencing these non-coding RNAs, for greater details, I would actually recommend you to go through our transcriptomics webinars. They are available on our website. So here I'm just briefly mentioning the library preparation types. So to capture the long non-coding RNAs, we do the ribosomal RNA removal, and the library type is directional. And this diagram shows what a directional library type is in terms of the experimental protocol. So directional library means that we are able to preserve original strand-specific information. It's also called strand-specific library because we can actually map or understand which strand is the sequence coming from. So basically, we have the original strand of our RNA. It is reverse transcribed. So we have this cDNA transcribed from the original strand. And then during the second strand cDNA synthesis, what we do is that instead of the thymine, the DTTP, we add the DUTP. And what we can do is that we can digest this DUTP later on. So the cDNA will be, which we'll have, is the one that is transcribed from the original RNA. And this is particularly useful for epigenetic studies. Now, also something I'd like to mention here is that in long non-coding RNA sequencing, we are doing the ribosomal RNA removal, which means that we'll capture the long non-coding RNAs, but we also capture the messenger RNAs. So when we do the long non-coding RNA sequencing with Novogene, you capture both mRNAs and long non-coding RNAs, which is beneficial for many cases. If you're interested in studying circular RNAs, then we again do the ribosomal RNA removal. The library type is directional, but we remove the linear RNA and enrich for circular RNA. If you're interested in small RNAs, then we do an 18 to 40 base pair insert small RNA library. Also, I'd like to mention here is that we accept pre-made libraries. Also, it's possible to do whole transcriptome sequencing, so you can capture all of these RNAs. And in that case, we'll have all of these different library preparation protocols. Finally, coming to the sequence analysis, sequencing data analysis of long non-coding RNA-seq. Like I mentioned, both long non-coding RNAs and messenger RNAs are included in the pipeline. So again, we have the raw data, we do the quality control, we map to the reference genome, then we identify the transcripts, so we can annotate the mRNAs, the long non-coding RNAs. If you have a number of samples or conditions, then the differential expression analysis is particularly useful. Based on that, we can also do functional analysis to see which pathways might be affected. And we can also offer you some structural analysis to maybe look for some alternative splicing sites and so on. Another thing I'd like to mention here is that because we don't have so much time today, I'm just showing you the long non-coding RNA pipeline, but we also have dedicated bioinformatics pipelines for circular RNA-seq, small RNA-seq, and whole transcriptome sequencing. With this, I'll come to the end. Just quickly summarizing the techniques we talked about so far. So we spoke about DNA modifications, primarily methylation, where we discussed bisulphite sequencing, both whole genome and reduced representation. We spoke about enzymatic methyl sequencing. And I also mentioned that it's possible to do methylation calls with long-read sequencing, with using platforms such as PacBio and Oxford Nanopore technology. We then talked about macromolecular interactions, protein DNA or protein RNA, which can be studied using ChIP-seq and RIP-seq. We also spoke about chromatin accessibility, which can be studied using the ATAC-seq technique. And we can also sequence non-coding RNAs like long non-coding RNAs or circular RNAs. And this is, again, providing us with a lot of helpful information to study epigenetics. So I will wrap up now, and I will take up questions. I know it was a short webinar, and it was to quickly summarize all these different techniques. If you have any questions, I would be very helpful. I would be very happy to answer them.
Yes, thank you, Arman, for this interesting talk and all the insights to epigenetics. So yes, we have a few questions, so I will read them for you. First question, can EM-seq differentiate between 5-MC and 5-HMC?
It cannot differentiate between the two. So the thing is that both 5-MC and 5-HMC are converted and are detected. So in the final sequence, you'll have both of them, but you cannot distinguish between the two. And that is a limitation. If you want to distinguish between the two, there are some tweaks to the protocol available. There are some experimental pipelines available that can help you to then identify 5-HMC. But if we talk about the regular method, then you cannot distinguish between the two, but you can detect both of them. I hope it makes it clear.
Great, thanks. A second one. What is the difference between ChIP-seq and ATAC-seq? A very general one.
Right. So ChIP-seq is to study these protein-DNA interactions, and ATAC-seq is for studying chromatin accessibility. Now, of course, there are differences in the experimental pipelines and so on, but in general, if we look at it, for ChIP-seq, there is a bit of bias in the sense that you need to know the protein of interest in order to immunoprecipitate it. So if you're targeting a particular histone protein, you need to have the information so you can use the appropriate antibody. While in ATAC-seq, you don't need this prior information. So that is a benefit of ATAC-seq, that you can go with less bias in that sense.
Okay, thanks. The next one. Can I send pre-made libraries for RRBS, so the reduced representation Bisulphite sequencing?
Yes, it is possible to send pre-made libraries. But I'd like to highlight here is that so you can do the enzymatic digestion and bisulfite treatment at your end, and you can make the libraries. But these libraries should be compatible with our sequencing platforms. So you need to add the appropriate adapters, the appropriate indexes. So in this case, actually, before you're trying to do the library preparation, maybe reach out to our account managers, because they can help you evaluate your library and to check if it is compatible with our platforms.
Great, thank you. Next question. What is the difference between DMR and DMP?
Right. So DMR is a differentially methylated region, and DMP would be differentially methylated position. So as the name tells you, DMR is for a region. So we're talking about a region of the genome. And for DMP, we're talking about a single site. So for DMP, we're talking about a single CPG site that maybe shows significant difference in methylation levels between two particular samples or between two groups or conditions that you have. While for DMR, we're talking about regions of the genome that show a significant difference in methylation between the samples or conditions that you have. Because we're talking about regions, maybe, the CPG-rich regions or the promoters, enhancers, and so on.
Okay. Then we have a last question for today's webinar. Can Novogene do an RNA joint analysis?
Possible to do RNA joint analysis. So we can study these RNA interactions. So we actually have a wide variety of joint analysis available, like long non-coding RNA, mRNA, long non-coding RNA, small RNA, mRNA. So there's a lot of variety that we can offer. So do reach out to our account managers, and they can provide you with more details. They can also show you some demo reports to see if the analysis fits your.
Yes, great. Thank you.
Thank you, Adrian. Any more questions? That's it. All right.
Yes.
So thank you all for joining. If you need more information, please send us an email, and we have a dedicated team, and we will be very happy to help answer your questions. Also, reach out to us and follow us on our social media platforms, where you get regular updates of our activities and the sequencing services that we provide. I'd also like to mention here that Novogene is now offering enzymatic methylation sequencing in Europe. So you can reach out to us if you have any projects. And we also have PacBio Revio platform in our new lab in Munich, in Germany. And as I mentioned, PacBio is also a great platform for epigenetic studies, primarily for doing direct methylation calls. So thank you again for joining us. And I look forward to seeing some of you in the next webinar.
Thank you also. Yes.
Thank you.